Hi guys, I hope every one of you are fine, safe, and preparing for your exams really well. Okay, so from today onwards, we are going to start Animal Biotechnology. Our planned biotechnology syllabus is over. So from today onwards, it's going to be Animal Biotechnology. Okay. So, what is animal biotechnology? So, animals are playing a growing role in the advancement of biotechnology as well as they are increasingly benefiting from the biotechnology. Combining the animals and biotechnology will result in four primary areas. One will be you will have advances in human health. Second one, you'll have improved animal health and welfare. Third, enhancements to animal products. Fourth, environmental and conservation benefits. All of this are the benefits of animal biotechnology. Also, animal biotechnology includes all the animals like livestock, poultry, fish, insects, companion animals, and laboratory animals. Okay, so the applications developed through research have led to the emergence of three scientific, agricultural, animal biotechnology sectors. So, the one is animal genomics, the second is animal cloning, and the third is genetic engineering of animals. These three are the important features and fields present in animal biotechnology, which is having high research applications. So the concept of animal tissue culture first was emerged in 1903 when scientists discovered the technique of dividing cell in vitro, which means the scientists started making the cells to divide in vitro. In vitro means outside your body in a test tube. So Ross Harrison made the beginning of animal tissue culture technique in 1907 using frog tissues. Okay, so he started using animal tissue culture technique and he used frog tissues. Okay, so this technique was initially confined to cold-blooded animals, but you know what is cold-blooded animals, right? And however, subsequent studies brought even the warm-blooded animals like us. We are warm-blooded animals. So over the years, various tissues have been used as explants. You know what is explant? Explant is nothing but your tissues which you obtain from animal for primary culture. Okay, so since we have already completed plant biotechnology, I at least hope you guys know what is explant and what primary culture and what tissue culture actually means. It is exactly same. Okay, so all these terms means the same both in plant biotechnology as well as in animal biotechnology. Okay, so using this explants and doing tissue culture techniques has become the backbone of animal biotechnology. And what are the benefits we get from animal biotechnology? One is that developing vaccines. Okay, so we can develop vaccines from the animal cell tissue culture. Next, you have role in animal breeding, embryo transfer, xenotransplantation, creating transgenic animals, improved nutrition. All of these are application in animal biotechnology. Okay. So, what is cell culture? Cell culture is basically a process in which you grow the cells in a controlled fashion, okay? In an artificial environment, in a controlled manner, you're just going to grow the cells. Cells can be maintained in vitro, okay? It can be maintained in vitro outside of their original body by this process, which is a quite simple compared to organ and tissue culture. Okay, so in a cell culture technique, cells are removed from an animal, and that is what is called as explant. Okay, that is what is called as explant or a plant and grown subsequently in a favorable environment. Okay, favorable environment because suitable environment. Okay, in suitable environment only your cell culture will grow. And it should be similar to the environment which is present in animals body. We cannot replicate it 100%, but yeah, to some extent it should be able to sustain the growth of those cells. For animal cell culture, the cells are taken from the organ of an experimental animal. So experimental animal can be your mice, it can be rabbit, it can be monkey, whatever it is, whichever experiment you are performing, that particular animal's organs cells are obtained. So the cells may be removed directly or by mechanical or enzymatic action. The cells can also be obtained by previously made cell line, which means the cells have been already obtained from the animal and it has been stored. Okay, it has been stored and you can use it anytime you want. So examples of cells used to culture are fibroblast, okay, lymphocytes, your heart cells, skeletal tissues, liver, breast, skin, kidney, and different types of tumor cells. All of these are done in cell culture. So, okay, so if you are growing the cell culture, it definitely needs environment. So, what are the environment that is required for the cells to grow? So, first one is the temperature. The temperature should be optimum such that your cells can grow well, and the temperature should be approximately equal to the temperature which is present in the animal's body. So, the suitable temperature and the optimal temperature is 37 degrees Celsius. Okay, so there are cells which will attach to the surface of your culture vessel and grow. And that attachment to the culture vessel is called a substratum. That is the substratum. Your culture vessel is the substratum and on which your cells will attach itself and grow. Okay, and next is your growth media. That is the most important things for the cell. As you need food for your growth and your development, the cells also need food, and its food is the growth media. Okay, carbon dioxide and pH, osmolarity, all of these things should be maintained paka. It should not be having any deviations at all. This is a standard thing, like 5 percentage carbon dioxide means yes, 5 percentage carbon dioxide only. So all of these things has to be maintained strictly so that you will get a proper yield. Okay, so next is your media components. Okay, what are the components that is present in the growth media? So you should have a carbon source, that is glucose. You should have a nitrogen source, your amino acids. Vitamins are required. Buffer system to maintain the pH. Serum will contain your growth factors, hormones for the mitosis process to occur. Then you have one antibiotic. You will also have antibiotic like penstrep, like penicillin and streptomycin. These antibiotics are definitely required because they will not allow the bacterial or any other contamination to come. Okay, then inorganic salts, attachment factors like substratum, all of these are important, and it should be like how the cells are growing in a person's body or any animal's body. Whatever is required for the growth of cell in an animal's body should be provided in vitro so that you will get a very good culture. Next is indicator. You will definitely use indicator in the media, okay, so that you will know what is going on, because these are cells. Okay, you can see only through the microscope, and you have to know like if they are growing or not, what is happening with them. Every time you cannot use the microscope, so just for your naked eyes and simple indications you should know. Okay, for that you're going to use an indicator in the media, and that is called as phenol red. Actually it is red here. Okay, phenol only is coming. This is phenol red. Okay, this is phenol red. Okay, so phenol red is used as an indicator in your culture medium. Okay, so this can actually change its color based on the pH. If the pH is 6.5, the color will be yellow. If the pH is 8.5, then the culture medium's color will become red. Okay, every color change is like an indication. Okay, every color change is an indication. Okay, now if pH is 6.5, then the media's color will be yellow. The media's color will be yellow, which means change of media is required as soon as possible, and maybe there is a chance of contamination. If pH is 6.7, then it will be like orange color. Okay, it will be like orange color. And what does that mean? It means that we have to change the media within a day. Within a day, you have at least you have a day of time to change the media. Okay, you should remove the already existing media and you should add a fresh media. That is what it means. Okay, next, if the pH is 7.4, then it will be red in color, which is perfect, which is like amazing, perfect. Next, if the pH is 8, then that will be pink color, which means carbon dioxide supply is less. Okay, so all of these are the indications. Okay, all of these are the indications. So based on the pH and based on the color which is forming, you have to take the necessary steps for the cell culture. Okay, all right. The more acidic it goes, the color becomes yellow, and the more basic it is, it becomes red and pinkish like that. Okay, so next is media. What kind of media you will use? If you're going to use natural media, then the components in the media is undefined. You do not know what are the components present in your natural media and its proportion. You do not know that. But yes, it does support your cell's growth. But it is not reproducible every time. So this is not preferred, because we are going to do the experiments in vitro in an artificial environment. Everything is artificial, so media is also artificial. Okay, media is artificial, and what does that mean? It means every component of the media is well defined, and its amount is defined, and how much you should use, when you should use, everything is properly defined. Okay, proper proportion. So if you have a proper proportion and it is optimized well, then you will get a reproducible result. Reproducible result means how many ever times you use the media, how many ever times you culture, you are going to get the same results, and that is what you want. If you are getting different results in different cultures, then that is a problem, right? Okay, so next you have a serum. So serum is nothing but the component of a blood which contains growth factors and hormones. All of this is very very important. Okay, all of this is very very important, because these growth factors and hormones are responsible for the growth of cells, for the cell division. All of these are used. So most commonly used serum in the cell culture is FBS. Okay, FBS is the most commonly used, that is fetal bovine serum. So if you're going to use a media that is DMEM media, that is Dulbicose media, if you're going to use it, you will use 10% FBS along with the Dulbicose media. Okay, so the serum contains mitogens. Mitogens are nothing but the agents which will induce your mitosis. Okay, that help in cell growth and division. Okay, so these mitogens are helpful in making of cyclin D. Cyclin D, what will cyclin D do? It will help in the transition of G1 to S phase. G1 to S phase transition, it will help. Cyclin D. So you also have serum free media. Some of the experiments do require serum free media, and for that you can use it. So since serum free media is synthetic, okay, it is synthetic, and knockout serum replacements are also available. Next, you will definitely use antibiotics in the medium, so that all of the components are well and safe from the bacteria, fungal growth. All of that should not be there. So you're going to use penstrep, which is like a penicillin streptomycin combination, and you'll use fungizone. All of this you'll use for bacteria and fungal growth. Okay. Okay, so primary cell culture. So what is primary cell culture? You know what is primary cell culture. We have already studied about all of this from the plant biotechnology. If you're going to obtain the explant from an animal and you're going to culture it for the first time, then it's called as primary culture. Okay, so you are obtaining the explant. Okay, it will be all the cells attached together with the cytoskeleton. You want individual cells separated from each other. For that, you're going to use trypsin. You're going to use trypsin. What trypsin will do is it will digest the cytoskeleton and you will get individual cells like this. You will get individual cells. You will get individual cells like this. So now you're going to take these cells, add media into that. So it will become a cell suspension. So cell suspension is nothing but it contains cells separated from each other along with your growth media. Okay, along with your growth media. Okay, cell suspension and growth media. So you know this is the growth kinetics of the cell, time and the number of cells. First they undergo lag phase, then log phase, then you have a plateau phase, or we call as stationary phase, plateau phase, and here the confluency is checked. And what is confluency? It means how much cell is present in your cell culture plate. Okay, that is confluency. Okay, crowding. So this is the stages. These are the different kinetics of the cells. So confluency is nothing but it is the crowding of cells. So if the confluency is more than 90 percentage, which means the cell culture plate is 90% filled with cells, then it requires subculture. And when you subculture the primary cell culture, it is either called as secondary cell culture or it becomes a cell line. Okay. So cell lines from the primary culture have the limited lifespan. Okay, cell lines which you obtain from the primary culture, they will have limited lifespan as they are passaged. Passage means subculture. When you subculture it several times, it will lose some of its properties and its lifespan will be reduced because the cells will undergo a process called as aging, or we also can call it as senescence. Okay, cells with highest growth capacities will be predominating and all of the cells will have genotypic phenotypic uniformity. Okay, so let's understand some of the differences between the primary cell culture and the cell lines. Okay, biological relevance, which means how much is it closer to the body. Okay, body cells. So primary cells are more relevant to your biological species, so it is high. Cell lines, they've undergone a lot of changes, a lot of mutations, so they are not at all like your body cells, so their biological relevance become less. Lifespans for primary cells is limited. Cell lines will undergo so many mutations and they become immortal in nature, so their unlimited growth. Next, consistency is medium consistency, high consistency, easy to use. Primary cell culture definitely requires optimization, but cell lines it is well established and optimized. Time and expense, primary cell culture is slow growth and cost is high. Cell lines fast growth because it is unlimited as well and it is of low cost. Okay, so these are some of the differences you should know about primary cell culture and cell lines. Okay, so how do you prepare a primary cell culture? Let's understand the preparation of primary cell culture with the help of a example. Okay, so we are going to culture hippocampus. Okay, hippocampus. So hippocampus is nothing but it is a portion of your brain which is involved into the short-term memory. Okay, it is involved in the short-term memory, learning, all of these things. Hippocampus is very important. So we are going to culture it. Okay, so brain, you're going to isolate it from the embryo of a mice. So mice which is pregnant, its embryo you're going to obtain, and from that embryo you're going to isolate the brain. Okay, now placing the brain in the ice cold PBS. PBS is phosphate buffer cell line. You know that is a buffer system. To dissect the hippocampus, first you isolate the brain, you keep it in the buffer system. Okay, then you're going to dissect the hippocampus and remove it. Okay, you are going to transfer this hippocampus in a solution that is containing enzymes like trypsin या फिर acutase or pronase. Okay, why all of these enzymes has the same function to remove the cytoskeleton from the tissues, you will get individual cells. all the cell types will be dissociated and you will get a suspension culture. Suspension culture means cells are all individual suspended in the media. You're going to incubate this culture along with the trypsin at 37 degrees Celsius for like 2 minutes so that all the cells will get detached from each other. You're going to pipette up and down to dissociate the tissues. You will get cell suspension. Next, you're going to count the cells. You're going to count the cells and you're going to take like 1 million cells or 1.5 million cells you're going to take. Okay, now you're going to take 1 or 1.5 million cells. And how are you going to count the cells? You're going to count the cells using hemocytometer. Hemocytometer is an instrument using which you can count your cells. After you count the cells, which should be 1 million or 1.5 million cells as an initial input, you are going to add media to those cells. Okay, you're going to add the media to that cell and resuspend it. Okay, suspension. Now you have to centrifuge it. You're going to centrifuge the cells along with the media. You have not removed this. You have not removed this um, trypsin yet. So we have to remove the trypsin. So what you are going to do is you're going to centrifuge. Okay, we're going to centrifuge. The supernatant will contain trypsin. You will remove it and you will wash the cells. Okay, now the supernatant which is containing trypsin is removed, but your pellet, which is the cells, will be present at the bottom of your tube. Okay, centrifuge tube. That pellet is your good cells without trypsin. Now you're going to wash those cells with PBS. Okay, so that if there is any traces of trypsin present, you can remove it. Next step is you're going to add the culture media and resuspend the cells. Okay, now you have a centrifuge tube which contains the pellet, PBS, the buffer and the culture media. Okay, now you're going to plate the cells in a culture vessel and you're going to incubate the cells. Okay, you're going to plate the cells in a culture vessel and you're going to incubate it. The cells will be present, media is present. Okay, so you're going to incubate at 37 degrees Celsius, 5% carbon dioxide. Okay, and you can change the media every alternative days because yes, the cells are growing, they're going to utilize the media, they're going to release the waste, everything will be present in the media. You're going to remove the media and you're going to add new fresh warm media so that they can utilize it. Okay, so you can change the alternative day media. Okay, you can change the media alternative day and you have to keep looking in the microscope for the confluency. If the crowding is more than 90%, go for subculturing. Okay, go for subculturing. Subculturing means from this you will take some cells and grow in a new culture medium. That is called as subculturing or passaging. Okay, so what is adherent cell culture? Adherent cell culture means the cells will be attached to the substratum. Substratum is your culture vessel. Okay, it is your culture vessel. Substratum. Regular passages are required for the adherent cells because they will grow in all of this region. Okay, after it grows to this region, it will stop growing. Okay, because they are contact dependent. If they feel it is so much crowding, they'll stop dividing. So you have to passage them regularly. Okay, they are easy to view under the microscope and it requires trypsin. If you're going to dissociate them, since they are attached, okay, they'll be like this, they'll be attached. Okay, like this. If you're going to dissociate them into individual cells like this, then you're going to use trypsin. Okay, growth is limited to the surface and which limits the product yield, which means it will just form a monolayer. Okay, it will form a monolayer and after that it will stop its growth. So the product will be limited to this growth. And this adherent cell culture has application in cytology, harvesting the products and research purposes. Okay, next is your suspension cell culture. So cells that does not adhere to the substrate like your red blood cells can be grown in suspension culture. They are very easy to passage. They does not require any trypsin. They do not require agitation or aeration. They are used for the bulk production of proteins and also for the research purposes they are used. Okay, so this is the diagram of your animal cell culture. This is your brain. Okay, you're going to remove the brain and you're going to remove some cells from the brain and you're going to culture it. And when the confluent cultures, you can see here, this is the culturing. Confluence means crowding. See, so many cells are present. When you find the confluency is more than 90 percentage, okay, 90 percentage, you're going to subculture. You're going to subculture, which is called as secondary culture. Okay, secondary culture. Next, it'll be you'll keep on subculturing. So cell division continues up to finite divisions. After that, it undergoes a process called as aging. Okay, aging or senescence. They'll stop growing. Okay, organ having a tumor, which means if your organ is having a cancer, okay, if your organ is having cancer, you remove the tumor or the cancer, okay, that is denoted as red color cells, and you transfer it to the culture vessel and you keep on subculturing. You keep on subculturing. They will not undergo aging or senescence. Since they're cancer cells, they will have indefinite cell divisions. Indefinite cell divisions, which means infinite, and that is called as cell line. Okay, either you can call your secondary culture as cell line, which is having finite divisions, or you can call your tumors or cancer subcultures as cell line, which is having indefinite or infinite divisions. Okay, I hope you understood the concept of what is animal biotechnology and how do you do your primary culture. Okay, so that is all for today, guys. If you are a CSI net aspirant and are interested in joining an academy platform, you can do that. Okay, you can do that by following Neha Taneja ma'am. You can do that by following Neha Taneja ma'am. And yes, what these are the pricings. Okay, these are the pricings. To get 10 percentage off, you can use the code Neha. You get daily live classes, structured courses, unlimited access, live tests and quizzes. All of these are present. Okay, so like, share, subscribe and also comment, so that if you comment, only when we will know what is your thoughts. So comment to our videos in the YouTube channel GurMantra Shiksha Ka Granth. You can join our Telegram channel where you'll get all the regular updates. Okay, you can join our Facebook page GurMantra Shiksha Ka Granth. You can also visit our website. If you have any queries or questions, you can contact us through the email and also call us if you want. So thank you so much, guys. Have a nice day and keep preparing well.